So before we conclude this genomics section, I just want to introduce some of the major projects that have led to very great leaps of advancements in genomics. So one of the most complicated genome project that was embarked on was the Human Genome Project. The idea was to identify the sequence of all the 23 chromosomes that make up the human genome. And then once you know the sequence of all the chromosomes, you will be able to identify all the genes, which is about, as I said, around 30,000, and also know where they lie on the chromosome. So now you can track them. The first human genome was published in 2002 and it made both Science and Nature paper, as you can see. The cost of doing that single genome was $6 billion. More than 1,000 scientists around the world in 50 countries were associated with it in doing the sequencing, and it took more than 10 years to do it. And as I mentioned earlier in the course, that nowadays we can do it in a couple of days. What was shown when the first sequence of the human genome came was that only 5% of the genome actually codes for proteins. Remainder is regulatory or of unknown function, or what is called junk DNA. We won't go into too many details of how do we identify these genes. Maybe that will be part of the course that is floated later. But the idea is that even though we have 3 billion bases, only a small fraction of those bases code for proteins. But we now know, as we discussed earlier, that there could be non-coding regions coding for proteins as well. So there might be a lot more in the remainder that we don't know, and a lot of work is continuously going on to understand this regulatory regions in the human genome. After this human genome was published in 2002, which was considered as a landmark paper, about 13 years later, there was another project that was published, which was launched actually in 6 years later after the human genome came, was the 1,000 Genomes Project. The whole idea was to sequence 1,000 healthy individuals from different ethnic groups so that we have a better understanding. Thank you. So the idea of this project was to sequence about 1,000 healthy individuals from different ethnic groups being individuals from different parts of the world. So because of their evolutionary trajectories, they would have different variation, so that we have a better comprehension of the total variation in the human genome. So to help refine the human genome map. And the goal were very simple, that by doing this 1,000 genome project, we will be able to identify rare variants which have large effects. We will be able to identify common variants which will have smaller effects on the phenotype but are involved in traits that are determined by multiple genes. And this was a resource that was developed almost 10 years ago and is still relevant today. So as you can see, by 2002 we had the human genome ready, the first draft of human genome ready, and in 2008 the 1,000 Genome Project was launched, and the publication of that happened in by 2013, and we are still using the human genome project data for many of our research at IIT Madras. Now we know also that Africa is the cradle for human evolution. The first humans were evolved in Africa, and then they migrated out of Africa and populated the rest of the world. So we do know that there is there is ample evidence to show that humans evolved in Africa and then colonized the rest of the world. So more ancient the population. So the idea is more ancient the population, more variation would exist in that population. So if we can imagine that since Africa was the cradle for human evolution, the human population, the population in Africa would be more ancient compared to the population in Europe or in Asia. And with that idea, African Genome Variation Project was launched in 2000, I think 18 or 19, where they sampled populations from different regions of Africa to understand how much is the variation that is existing in the population of Africa. And you don't really have to read this graph right now, but this study actually is very important because it gives you the list of variants that are more ancient than the list of variants that would have been identified in European populations. And therefore, now with this African Genome Variation Project, we have not only variants that are more ancestral, so that they would be common across most of the population, but also variants that do not exist anywhere else in the population outside Africa and are very unique to African population. Finally, I would point you out to a very large resource. This is one of the biggest of the big data in biology, which is called the UK Biobank. It's a large scale biomedical database which has both genetic and health information of almost 500,000 UK participants. And one can actually access this information for research purposes. So what does it have? It has genetic data, it has physical activity data, it has a lot of surveys, it has a lot of images which could include both X-rays and MRIs, it has lot of samples that have been characterized, it has a lot of mental and cognition data sets, what were the lifestyles of all of these 500,000 individuals, and what are called anthropometric measures, weight, height, various other physical features. And this has become one of the most comprehensive data sets available to researchers to test and develop hypotheses. And this biobank, UK Biobank, is one of the major contributors to development of several modern medicines and treatments, and is continuously being upgraded with more and more data which can then allow more comprehensive understanding of many diseases. And so as I said, the future goals are to get more data on how these individuals are, so it's becoming more and more longitudinal. So they have already this 500,000 participants, they're tracking them continuously to see what is happening. So now you have not only have genetic information, you also know how that genetic information leads to what diseases, right? So that is another thing. They are also sequencing the whole genome, right? This genetic data is sort of targeted, but they also are adding more and more genetic information. So it would in future be one of the most comprehensive data sets available for biomedical researchers, for computer scientists, for computational biologists to develop algorithms to learn from this huge big data, to identify biomarkers, identify genes that are involved in various diseases, and also do predictive analysis based on the longitudinal records that are available in the data set. Finish. The ending part again. So by covering these topics in the genomics, scrap. So as you can see, genomics not just gives the sequence information, it has a lot of utility in identifying disease variants, identifying disease genes, identifying genetic causes of various diseases, and also helping develop various hypotheses which can be tested further. So genomics is a vibrant big data set which is being currently utilized to do a lot of biomedical research. Next, what we will cover is how transcript level information, which is transcriptomics, which is the first readout of the DNA, is being utilized for uh, identifying gene expression changes, gene expression networks, etc., etc.